HPV is a group of small icosahedral non-enveloped DNA virus that specifically infect epithelial cells. There are over 100 different types of HPVs, which can be categorized by the epithelial cells they prefer to infect. HPV is classified into two categories, which are low-risk HPVs and high-risk. This is the HPV genome structure. It consists of about 8 open reading frames that are divided into 3 functional regions: the early region, the late region, and the long control region, which is a non-coding part. The genes, which are E4, E5, E6, and E7, play roles in cell entry, immune evasion, and virus release, thus involve in the formation of koilocytes. There are 3 stages of HPV infection. First, it starts with early stage of infection. Next, the differentiation and maturation of HPV-infected cells. Lastly, late stage of infection. First, oncoprotein E6 binds and promotes the degradation of the tumor suppressor protein P53. Normally, P53 helps prevent abnormal cell growth and initiates apoptosis, which is cell death. However, inactivation of P53 inhibits its normal functions in cell cycle control and apoptosis. This interference with P53 can impact keratinocyte differentiation, as P53 is involved in regulating genes responsible for proper cell maturation. As virus prevents host cell from undergoing apoptosis, it will allow the cell to persist and replicate. For oncoprotein E7, it binds and disrupts the function of PRB, retinoblastoma protein. So PRB hormonally inhibits the activity of E2F transcription factors, which are critical regulators of cell cycle progression and differentiation. E7 then promotes the release of E2F, allowing for uncontrolled cell division and potentially affecting keratinocyte differentiation. As a result, HPV-infected keratinocytes may exhibit a retention of immature characteristics and fail to properly mature into fully differentiated cells, which bring us to the second stage, differentiation and maturation. As infected basal cells progress through the process of differentiation and maturation, they undergo changes in gene expression and cellular morphology. The infected cells move towards the upper layers of the epithelium, where they continue to replicate and produce viral proteins. In the late stage of infection, the E4 and E5 will continue the role of forming koilocytes by disrupting cytoskeleton and cellular membrane. Cytoskeleton consists of microtubules, fibers, and filaments. At this site, the fibers and filaments can still adhere to the cell and maintain the cell shape and structure. After E4 and E5 attacking the cell, changes occur where the microtubules will be shortened and will be more curved and bent. They are regularly localized more perinuclearly. Actin stress fibers are reduced in number, length, and thickness. This caused the rearrangement of cytokeratin and increase of distance in nuclear. Then it will be resulting in cytoplasmic vacuolation and nuclear displacement. Intermediate filaments form clusters, and larger meshes appear in the network, and the localization is more perinuclear, which we called as coilocytes. Next, the inflammation that lead the formation perinuclear halo. First come from inflammatory mediator that lead to induction of oxidative stress and activation of stress response pathway. Lastly, perinuclear halo form. Inflammation condition can cause by infection, tissue injury, and autoimmune. In this cases, the inflammation caused by the cell that infected by the HPV. Neighbor cell becomes stressed with the viral infection situation. This inflammation caused the activation of immune response such as macrophages and lymphocytes. Macrophages then release reactive oxygen stimulates while the T lymphocytes release cytokine. These two substances can cause a condition called as induction of oxidative stress and activation of stress response pathway. In induction, oxidative stress causes increasing number of reactive oxygen stimulate. It's caused the oxidative damage toward the DNA. This nuclear alteration also somehow can cause nuclear envelope alteration. Lamin and nuclear pore complex are two examples of nuclear envelope components that being altered. Function of lamin is to protect the nucleus structure. But then, when it's being altered, there will be rearrange of lamin that cause formation of gap or space around the nucleus. Second, for nuclear pore complex, is to transport molecules from nucleus to cytoplasm. But when it being altered, there will have nucleocytoplasmic changes. The molecules will be accumulated around the nuclear region. That's how perinuclear halo is formed. For endoplasmic reticulum stress, it will happen when there is an imbalance between protein folding needs and ER capacity to handle them. This can happen due to a lot of factors, including viral infections or misfolded proteins. Because of this, unfolded protein response UPR will be activated, but if stress persists, it can cause cell dysfunction and apoptosis. During ER stress, the UPR can change nuclear shape, size, and chromatin organization, potentially resulting in perinuclear halo. Severe or prolonged ER stress can trigger apoptosis, leading to nuclear fragmentation and the formation of apoptotic bodies. These changes, together with altered chromatin organization, can contribute to perinuclear halo formation. For Golgi apparatus disruption, the disruption of Golgi apparatus can affect cellular component and processes related to nuclear organization and chromatin structure. It can be caused by factor like viral infection, genetic mutations, or drugs. This can lead to impaired protein trafficking and modification. It can affect the function of nuclear proteins involved in chromatin organization, leading to changes in nuclear structure and formation of perinuclear halo. So what is the difference between coilocyte and perinuclear halo? Koilocytosis is a descriptive term derived from the Greek adjective kilos, meaning hollow. While for perinuclear halo, peri comes from the Greek word which means around and surrounding, and halo term refer to circular or ring-like structure. The formation of coilocyte occurred due to viral infection, which is human papillomavirus, HPV, while the formation of perinuclear halo can occur because of cellular stress and metabolic changes due to HPV infection. The other cell characteristic for HPV infection is enlarged and irregularly nuclear shape, nuclear hyperchromasia, abnormal chromatin distribution, nuclear atypia, granular cytoplasm, and vacuolization.